Lecture number 3 is going to cover the anatomy of a scientific peer-reviewed journal publication. However, I just want to review a couple of quick concepts before we dive into that material. At the end of the last lecture, we talked about this 7 parts to designing a research project. And so I just wanted to refresh your memory with this again. So when we are designing a research project, the first thing we do is ask some sort of question and then construct null and alternative hypotheses. Then we'll get into identifying independent variables, the traits that are going to be measured, what data we want to collect, how we're going to collect the data, and then start assessing what our expected outcomes would then be. And so as we dive into new material today around the concept of really taking apart a piece of peer-reviewed literature, I want to make sure that we have a handle on some terminology. We briefly introduced these to you in previous lectures, but now is the time to commit them to memory. So the first new term that you need to make sure you have a handle on is independent variable. And so this is going to refer to the unit or the manipulation point or the treatment that is going to be applied to the experimental unit. Okay, so a lot of times we call independent variables treatments. And just keep in mind that an independent variable will also include a control. So for example, if we are looking at applying two different fertilizer levels to a field of alfalfa, and we are going to have a plot study. Part of those plots are going to get treatment number 1, which would be like a high fertilizer level. Part of the plots are going to get treatment number 2, which would be lower fertilizer levels. And then we would also need to create plots that would be treatment number 3, which would be no fertilizer levels or a control. So it's really important that we always create treatment groups that are going to have a baseline measurement, and those usually end up being our control treatments. So keep in mind that a treatment or an independent variable is going to be a variable that is going to be manipulated. The dependent variable is going to be the trait that is going to be measured or is going to respond to what is being manipulated. So, for example, if we go back to the alfalfa example we just talked about, a dependent variable could be tonnage, or it could be plant height, or it could be number of leaves on a plant, etc. Okay, so those traits are going to depend on the treatment that was applied to them. The third term that we need to make sure we have a good handle on is experimental unit. And this is you're gonna hear this term come up over and over and over again, but it is the single most unit that a treatment can be applied to. So it could be an individual plant, an individual animal, it could be a whole entire plot, or a whole entire pen. The concept of randomization was introduced at the end of lecture number 1, but it really refers to how the experimental units are going to be assigned to their specific treatment. So that needs to be as random as possible. And the reason that randomization is a core principle of experimental design is because randomization will ensure an equal chance of an experimental unit being assigned to each treatment. And so this randomization act removes the potential for bias as well as removes extraneous or uncontrollable variation. And variation is the spice of life when it comes to research. So we're going to talk about variation a lot. The second principle of experimental design was replication, and this is the repetition of an experiment. So how many times an experiment is replicated. And replication is really important because it will eliminate that variation that we see between experimental units. Okay, so we can actually decrease the number of variation that we see between experimental units by having more experimental units and more replication in a study. So this will help us obtain really accurate measurements of experimental error, or how effective our treatments are actually being. And so this helps to increase the precision of a research project. And finally, the last term to make sure we have a good handle on is the third principle of experimental design, which is local control. And so this is the measures that are going to be put in place to balance, block, or group experimental units that are used in a study. And so the term control means or refers to a group of experimental units that do not necessarily receive a treatment. And so those local controls are really, really important because it allows us to compare the other treatments that we are interested in to a base. All right, so now we can dive into the anatomy of a scientific publication. And if you guys remember back to the very first set of lectures, I showed you this timeline of the scientific method, where we start out with observing a problem and we end with a scientific publication. And so scientific publications are the coup de grace, or the end point of the scientific method. It's really important that we remember we have to communicate our results. And so that tends to be the end point of the scientific method. And so we are very much beginning this course with the end in mind. And that's why we're talking about the anatomy of a peer-reviewed paper at the very beginning of class. And another big reason is because in order to do the scientific method process due justice, we're going to have to leverage scientific publications clear back in the very beginning part of the scientific method when we do our literature review. So it's important for us to do a good job with a literature review because we know and we can maneuver our way through a peer-reviewed publication. So let's start pulling these pieces apart. So the general parts of a scientific report that you will always see is a title, an abstract, which you can think of as kind of a summary. Okay, there will always be an introduction, which is going to share background and justification for why the project took place. It's going to introduce the question that was initially started down this pathway, and it will also introduce the hypotheses. So usually the alternative hypothesis, sometimes the null hypothesis. And then there will be a method section, and this is called a lot of different things depending on the journal. So in Journal of Animal Science, it's called Materials and Methods. In the proteomics journals, it's called Methodology, and there's a lot of variations in between. But it is that part of that scientific report is going to outline what methods were actually carried out in the study. The results section is going to report what was found in the study. Okay, so that's going to be reported in text, through graphs, through tables, and it's going to summarize the data from an analytical standpoint. The discussion section, as we previously mentioned, may come in combination with the results section, or it may be a standalone section of the journal. It just depends on the publication that you're looking at. But the discussion section is really important because it's an opportunity for the researcher to compare the results of their study with what has already been published in literature. It's also an area for interpretations to take place and predictions, as well as next steps. And then at the end of a scientific report, you'll always see a section for acknowledgments. So it's important to acknowledge people that helped in the process along the way, even if they weren't included as an author. But it's also even more important that you thank people that gave you money to carry out the project. So this is an area where you can acknowledge funding agencies like the USDA or an extension report or a private donor. And then last but not least is references. And so for all intents and purposes in this class, we are going to use the APA citation method for references, which is fairly standard throughout most peer-reviewed journals. There's a few of them that have different variations of APA, but for the most part, all that information that you would find in a citation in an APA format, like the authors, the year, the title of the paper, the where you found it, the DOI, all that good stuff is going to be the same. The order and the format just varies a little. All right, so as we look at the anatomy of a research paper, we're going to spend most of our time talking in this lecture talking about the title, the introduction, materials and methods, results and discussion, conclusions or implications, and literature cited. We're going to save the talk about the abstract to the very end of class because that's really a summary, and there's so you have to generate all this other information first before you could even write an abstract. And so typically we're taught to write an abstract at the very end of pulling a peer-reviewed manuscript together. It doesn't always happen that way. I'll share my experiences a little bit later on. But this is what we're going to spend quite a bit of time talking about in this lecture.